Unfortunately, after that, things started going rather more poorly. The next gene that they tried, the same group, actually, same research group, couldn't get it to work. They moved the gene, but they couldn't get the gene to actually function properly. And lots of people essentially lost hope after that. There was enormous interest in this for a few years around here, but that interest waned as people hit problems and couldn't get the thing to work, especially not in mammalian cells. An absolute hero named Steve Zulo succeeded in 2000, after an enormous amount of perseverance and basically a bit of luck, being able to essentially do a lot of work in a lab where he wasn't really being paid, he got one gene to work. And that was a major breakthrough. Here is a graph from the paper that he published in this, about this. And essentially what's happening here is these two dark lines up here are control situations where you've got cells being grown in a medium that allows them to grow even if they can't do oxidative phosphorylation. So this is the set of cells that do not have the transferred gene in their nuclear DNA, and also the ones that do. Then this line here that trails off and becomes horizontal, in other words, growth of the cells stops, is cells which do not have the transgene, the gene that has been modified and put into the nucleus, and they are being grown in medium where they do need to do oxidative phosphorylation in order to grow. Okay, so as you can see, they grow for a little while and then they just stop growing. The important line is this one here, which is cells being grown in that non-permissive medium where they can't grow unless they can do oxidative phosphorylation, but they've got this transferred gene. And as you can see, they're doing fine. They take a little while to take off, and we think we know why that is. But once they do take off, they grow almost as well as cells that are being grown in the permissive medium. So that was a massive breakthrough. But there's a lot that we still don't know about quite how they got it to work. They may have been just very lucky in a bunch of ways that we don't understand yet. So what's the deal? I mean, this sounds like a simple concept, as you can see. You know, you've basically just got to do some fairly straightforward modifications to put a gene into the nucleus in a manner that means that it ought to work. What's actually really the problem? It's this problem. One word explains it all: hydrophobicity. What is hydrophobicity? It is a property, a measure of how much proteins try to essentially get away from water. Hydro for water, phobicity for fear. Okay, so a protein that has a lot of hydrophobic amino acids in it tends to fold up into a ball so that it minimizes the contact that those amino acids have with water. The amino acids that are hydrophobic tend to be surrounded by other amino acids so that water can't get to them. And they do this, just they fall into a low energy state in that way. Why is that a problem? Well, it's a problem because a protein that it wants to fold up into a ball takes a lot of energy to unfold. And it turns out that the machinery that imports a protein into the mitochondrion from the cytosol absolutely requires to unfold that protein. So these 13 proteins, it turns out, these 13 proteins that are encoded in the mitochondrial DNA are viciously hydrophobic, and they really, really don't like to unfold. We now believe that the only reason that that Australian group had such success in the 1980s were because they were working with such a small protein that by just, pulling really hard, the mitochondria were able to get the thing to work. But with a bigger protein, it just doesn't work. It just doesn't happen. So this seems to be the only real difficulty. So there are some cases where hydrophobicity doesn't seem to matter too much. First of all, a lot of people look often at chloroplasts, which of course we don't have. They exist in plants. But they're interesting because they also have their own DNA. It's very similar in many ways to the mitochondrial DNA. And people are interested in comparing the two. Now, it turns out that chloroplasts have quite a lot of genes that they encode in their DNA, that the proteins are not particularly hydrophobic. But also, there are proteins in the chloroplast that are really hydrophobic, and they get in okay. They seem to be imported without difficulty. But we know why that is, because actually this thing that I told you about with regard to having to unfold the protein in order to get it in isn't true in chloroplasts. They don't have a problem because essentially they don't do an important aspect of oxidative phosphorylation in a membrane that surrounds the DNA. There are other examples in the mitochondrion. There's a group of proteins called the anion carriers, which are really hydrophobic, but which also are encoded in the nuclear DNA. and they seem to be imported okay. But it turns out that the way that they're imported has evolved in a different way, and it's completely different, and for reasons that I don't have time to go into, we can't use that route to get these 13 proteins in. There's another little hint, however. It turns out that you can make a protein that's got one really, really viciously hydrophobic slice in it, and it gets in okay. Okay, so long as there's not too many of these hydrophobic domains, as they're called, little substrings of the amino acid sequence, you don't seem to have a problem. And that's pretty interesting. And we think we know what's going on now. A phenomenon was discovered about 35 years ago in yeast called co-translational import, where essentially what happens is that the proteins that are imported into the mitochondrion are imported while they are still being synthesized. The import process begins before synthesis has really proceeded very far. And this turns out to be a rather interesting phenomenon. First of all, we don't know how it works, really. We do find that it happens with a lot of these proteins. What's so interesting about it? Well, the interesting thing is, if you think about it, it sidesteps the hydrophobicity problem completely. Because what it does is it causes the import process to happen before the protein has any opportunity to fold up the way it likes to do, being hydrophobic. So this is basically the concept. You've got the standard concept of protein import, where the protein is synthesized on ribosomes attached to the messenger RNA, and then after that the protein is targeted to the mitochondrion, and it gets stuck and it can't get through because it won't unfold. But then you've got this alternative thing where the messenger RNA is sitting on the outside of the mitochondrion being translated, and the protein is going straight in before folding up. A couple of years ago, a group in Paris made a massive breakthrough. I said that we don't know how this works, but we do know one thing now about how it works. We know that it's controlled by a region of the messenger RNA at the back end that doesn't actually encode any of the protein. It's called the 3' untranslated region. And it's at the far end of the messenger RNA, and because it doesn't encode any amino acids, you can stick that sequence onto the back end of any protein you like, any coding sequence you like. So you can end up with a gene that encodes a protein that can be imported, even though the protein itself is hydrophobic. And it turns out that it works. This seems to be a more or less complete solution to the hydrophobicity barrier. We've got this situation now. This is data that was published by this group a couple of years ago now, well, a year and a half ago. And it's the same sort of deal, same sort of four growth curves that I showed you in a different context from the Zulo group from a few years ago. These two dark ones are the controls, where the cells are growing perfectly well because they're growing in a medium where with glucose, where they don't need oxidative phosphorylation. And this bottom line is the cells which do not have the new gene, and they can't grow in galactose, which is the non-permissive medium. And sure enough, this one is what matters, the cells growing in the non-permissive medium, but with the transgene, and they're perfectly happy. So we're pretty happy about this. Mutations do accumulate during aging. They may be really important. We may be able to make them not matter, to obviate them, by putting copies of those genes, suitably modified copies, into the nucleus. And we have, maybe some way to go to get the whole story working, to get all 13 of these proteins working at the same time in the same cells, but it's going pretty well. Of course, after this, we've then got we can only use this therapeutically if we can get those genes into our cells of people who are already alive. And for most of the types of cell that are affected by this problem during aging, that means we're going to have to develop really good, safe, and effective gene therapy, which, as all of you know, is still some way away. But even that is an area in which there is considerable progress at the moment. So we have reasonable cause for optimism. And the sooner we push this forward more energetically, the sooner it will actually happen. So I'm going to stop there on this talk. I think that's, it's not going forward anymore, so that must be my last slide. And I'm happy to stop here and answer questions for a few minutes.